Hey guys, it's Nora. In today's video, we're going to talk about using the Pluto platform to do two things. The first is single-cell RNA-seq or single-nucleus RNA-seq analysis, and the second is scientific storytelling using the Pluto Canvas. I'm going to do this all in the context of a paper on non-alcoholic fatty liver disease. So one of the really interesting features of the liver is that it's demonstrated the ability to regenerate following acute injury. But the processes, the actual biological processes that underlie that regenerative ability are not well understood. So the authors of this paper set out to establish a cellular map of the liver throughout the disease progression in order to better observe and characterize these regenerative processes. And the methods that they used to do that were imaging and then also single-nucleus RNA-seq analysis on 47 liver biopsies from patients that spanned the entire progression of NAFLD. And they had some pretty interesting observations, the most important of which, or the one that I will highlight, is that they observed transdifferentiation events between hepatocytes and cholangiocytes without the presence of stem cells or developmental progenitor activation. And we're going to get more into that finding later on in the video. But before we do that, I want to start by just talking about how we actually analyze this data using the single-cell modules on the Pluto platform. So after pulling in my FASTQ files, I used Pluto to run the NF-core single-cell RNA-seq pipeline. That pipeline uses CellRanger to convert raw FASTQ files into a count matrix that contains the number of transcripts observed per gene in each cell. Out of that pipeline also comes a QC report where we can observe things like the number of valid UMIs per cells, the estimated number of cells per sample, and other useful QC metrics like that. I then took my counts data on into a pre-processing workflow. The pre-processing workflow calls scRNA commands and performs several steps such as filtration, normalization, data integration. But what's really useful here is that I was able to basically exactly reproduce the workflow that the authors did. So for example, I'll go over to the paper and we see in the methods section that they've listed, as good scientists do, the exact parameters that they used for filtering. So for example, to remove potential doublets, they eliminated any nuclei with more than 50,000 UMI counts. And that's something that's very common, is eliminating cells that have way more than expected numbers of UMIs to eliminate any events where potentially two cells were basically sequenced together. So I was able to take that parameter and then open up the filter module, and I have control over the thresholds for filtering strategies for UMIs, number of features per cell, complexity, mitochondrial DNA content. So I just set the maximum number of UMIs per cell to be 50,000 to match what the authors did. I also noted that the authors used the normalization strategy of scTransform, so I was able to set that in my normalization module. So this is just a really useful tool for being able to do the entire single-cell workflow by yourself and reproduce the analysis that might normally be done in R using scRNA, but entirely through a graphical interface. As an aside, I think single-cell RNA-seq analysis can be pretty daunting, and each of these steps can feel like a black box in its own way. If you're interested in a more complete single-cell RNA-seq how-to workflow that really breaks down what's going on in each of these steps in depth, please leave a comment below, or you can email me, and I would love to produce that kind of video for you. So once we're done with pre-processing, we can move on to annotation. By default on Pluto, we execute clustering with 4 different resolutions. And it's important to note that the higher the resolution you set, the more clusters are going to be produced. So that's something where you can put your biologist hat on and kind of think about the number of cell types you're expecting to see. And if you don't expect to see 100 clusters, then maybe decrease your resolution. So I'm going to click on this first clustering, and I'm going to show how you can do annotation with 2 different methods. So here you can click on any specific cluster. So I'm going to click on this dark blue cluster here, and you'll see all of the marker genes that are observed in that cluster. So those are genes that are differentially expressed in that cluster relative to the other cells in the plot. So you can use these marker genes that you are expecting to see from a specific cell type and then label the cluster manually. So for example, if BIC1 is your marker gene for cholangiocytes, then you can label it accordingly based on expression of that marker. However, cell type annotation can often be a bit more complicated than that. So there's a nice feature on Pluto. So you can click Help Identify this cluster, and then we use CellKB to generate cell type suggestions for you. So you can select your organism and then set a few options, such as the tissue type. So here we know that we're working with liver. You could set a disease type. I have patients at varying degrees of disease progression, so I'm going to leave that blank. And then you could even put in cell type. So I could put in cholangiocytes, hepatocytes, lymphocytes, things of that nature to help it kind of narrow down what it's looking for. And then I'll click Load Cell KB Suggestion. Cell KB is then going to return to me its best matching cell types. So I can see cholangiocytes here have a high score and a low false discovery rate, and that's what you want to see. Is the higher the score and the lower the FDR, the better the annotation. And I can also see some of those marker genes that I was interested in appear here in the overlapping canonical markers. So I'm going to select Use as Label, and then I can X out of this. If you are new to the world of single-cell RNA-seq, I do want to briefly mention what exactly these plots mean. So on the left-hand side, we have a UMAP. Over in the right corner, we have a t-SNE. They're basically two different flavors of doing the same kind of analysis. But on this plot, the axes are arbitrary. And what's important is the distance between clusters. So on this plot, each point represents a single cell, and distance between points is a proxy for similarity of expression profile. So all of these points in this blue cluster are all cells that have very similar expression profiles, and they are different than the cells that are in this kind of pink colored cluster, which I believe are lymphocytes. But all of these cells that are grouped together in this big mass are, relatively similar, and those I collectively grouped as hepatocytes. But those clusters, many of them originate from different patients. So we have healthy patients represented in that population as well as end-stage disease patients. This kind of plot is really a bread and butter analysis for single-cell RNA-seq, so I just wanted to mention that briefly. And now that we've done cluster annotation, we can move on to analysis. So let's recall that the question the authors are trying to answer here is, what are the processes that underlie the liver's ability to regenerate? So the first analysis that I did was just to color my cells according to disease status. And what we see here is that cell type specific clusters mostly overlap regardless of their disease stage, with the exception of the hepatocytes, which do display clear transcriptional changes according to disease progression. And this is confirmed by the plot on the left, where cells are colored based on cell type. We note that even with multiple disease states combined, there's still really only one cholangiocyte cluster, one lymphocyte cluster, and so on and so forth, but there is quite a bit of heterogeneity amongst the hepatocytes. So I've labeled them all and colored them the same just for ease of visualization, but this big group of hepatocytes here represents hepatocytes from many different disease conditions, and we do note that they kind of break out into distinct clusters. So it makes sense that the most transcriptional changes during disease progression are observed in the hepatocytes, because the hepatocytes represent the primary cell of the liver and are known to be most affected by NAFLD. So one of the most interesting changes that the authors observed in their scRNA-seq analysis was the presence of cells that co-express hepatocyte and cholangiocyte markers. So they took two markers for cholangiocytes, which is KRT7 and BICC1, and they observed that these markers were also expressed in the hepatocyte clusters. And then the marker for hepatocytes, ABCC2, was also expressed in the cholangiocytes. They confirmed this more strongly with imaging of different markers. So they used albumin as a hepatocyte marker and K7 and K19 as cholangiocyte markers. And there's a lot happening in this figure, but I'll just call your attention to figure B, where you can see in row 2, they've stained these end-stage hepatocytes for K7, and then in row 3, they've stained for K19, and in the last row, they've stained for albumin. But across the top, you can see all three kind of overlaid on top of each other, and you see that there are these biphenotypic cells that express both cholangiocyte and hepatocyte markers. So this led the authors to believe that there's basically an increase in plasticity during disease progression, or at the end stage of liver disease. So one of the analyses that I did was differential expression between clusters using the Pluto platform's differential expression tool. So I compared end-stage cholangiocytes versus healthy cholangiocytes and end-stage hepatocytes versus healthy hepatocytes, and I noted an enrichment in one of the plasticity factors that the authors looked for called KLF6. So these findings basically provided evidence for a process called transdifferentiation. So transdifferentiation is a process where one differentiated cell type transforms into another differentiated cell type without passing through a pluripotent or progenitor stage. And it's important to note for the context of this paper that transdifferentiation is a regeneration process. So if you think about how you could, generate a cell that is lost, one way to do that is for a already differentiated cell to convert to that cell type. And if you are a Harry Potter fan like me, this immediately made me think of Transfiguration and Professor McGonagall. So the authors then wanted to understand what mechanism causes this transdifferentiation to occur. And when they did a gene set enrichment on the genes specific to these biphenotypic cells, it indicated an enrichment in Go terms for processes associated with mTOR, PI3K, AKT signaling, which I will now just refer to as mTOR signaling because that is quite a mouthful. But they used intrahepatic cholangiocyte organoids, and they exposed those ICOs to mTOR pathway inhibitors. So they had an mTOR inhibitor, which is ripamycin, they had an AKT inhibitor and a PI3K inhibitor, and they noted that when the ICOs were exposed to these inhibitors, that they displayed much lower expression of hepatocyte markers. This was also confirmed with imaging, so you can see that the ICOs that are in the presence of the mTOR inhibitor ripamycin demonstrate reduced hepatocyte marker expression. One of the things that the authors note is that the mTOR pathway is activated by the presence of insulin, and in-stage liver disease patients often show insulin resistance. So taken together, these findings could indicate that the increased circulating insulin during liver disease progression may have a key role through the PI3K, AKT, mTOR pathway in inducing the plasticity in the hepatic epithelium that causes this transdifferentiation. Before we close out the video, I do just want to zoom out for a second and show you the kind of complete story that we've told through the Canvas. I personally find this to be a really helpful tool for pulling together all of my figures and my notes and my visualizations and really figuring out the story that I want to tell with my data. If you're interested in testing out the Canvas or trying out single-cell RNA-seq analysis on the Pluto platform, we would love to speak to you. Please just contact us at the link below in the description, and I will see you in the next video. Thanks so much for watching.